First, gram staining. I hope you getting it what I am saying. Okay, gram staining. It helps. It helps to distinguish between gram positive and gram negative. Okay, so why we are doing gram staining? So all thing works in process. Okay, first you do gram staining, so you know which is gram positive or gram negative. Okay, so if you know the gram, it is gram positive, then you go for the catalyst test. So you know that we have cocci in a smear microscopy, and you found in clusters or you found in chains. There is also a different method of distinguishing staphylococcus from streptococcus. Okay, but this gram staining helps to distinguish between gram negative and gram positive. Okay, it is a process containing about 10 steps. 10 step. It is a very easy process, and also it's it's quite sufficient to help us to distinguish between them. And first of all, we will do this. Then we go for catalyst, so we can differentiate. Then for the species of staphylococcus, we have different tests. So this lecture will be based on this test, and all the tests we will explain one by one, and what are the characteristic of all the species by this test. Okay, so in gram staining, you are in a lab. Okay, just suppose you are in lab, and here it is a table. It is a table. Okay, this is a table. My drawing is not good, it is bad. So here is a table, and here at the end there is a sink with tap water. Okay, you can see this is a sink with a tap water, and you have a rack. What it means a rack? With a parallel rods. Okay, it is a rack with parallel rods. So parallel rods are placed in a as a rack. Okay, so what you have to do, you have to take a slide. You have to take a slide. Okay, this is slide. Okay, so we have to place this slide perpendicular to the rack. Understanding what I am saying? You have to take a slide and put a perpendicular to the rack. Now this slide contains a smear. Okay, you have to examine this smear, and to examine this smear under microscopy, you have to perform some chemical reactions. Now you have to add some solutions for it. What solution we will basically use in whole gram staining procedure I am writing here. First of all, you will use violet, crystal violet stain. Okay, crystal violet stain. And second, you will use iodine solution. Third, you will use acetone for decolorization. Fourth, you will use saffron saffron, sorry for my pronunciation, earlier told you, saffron solution. It is a counter stain. This is a counter stain. This is orange or like that color, saffron. So it's, the word has derived from that. And acetone is a decolorization agent. Decolorizer. So you can write decolorization, and iodine is about yellow, and violet is about purple color. So we will use, so we will perform this activity with the help of these four agents. May I
think so. So this is the stain, crystal violet stain. Earlier we talked about the stains. There are four stains: crystal violet, saffron, iodine and acetone. So it is one of the main stain we are going to use at the as a first step on the smear containing slide containing a smear. So this is the crystal violet stain. You can see the color and the depiction about the image. You can get it and have a clue about it. So we are going to the second image. This is the iodine stain used as a second reagent after crystal violet stain, and we have to keep the slide for 2 minutes after installing the fresh iodine solution on the smear. Okay, so this is the second stain. You can also call Lugol's iodine solution or Lugol's solution. So yeah, we are going to the third image. Yeah, this is the rack what I was talking about. The rack is containing two parallel rods which are placed perpendicular to the slide, and the slide is placed horizontally, so the it's easy for installing or for staining the slide, and it's quite possible by this geometry. It's easy for the lab technician to install the stains and stain the slide and do the further procedure of gram staining. And you can also see in this image there is a sink nearby this rack, so it's easy for the tap water to run down the sink, and there is no spillage of any of the stain out of this rack, out of this sink. Okay, so it's done near the sink. So here we go for the fourth image. This is the image I will talk about in later video, in this video only, but at the last moment, because it's done after gram staining, and this is the smear microscopy results, so we are going to talk and later. And this is also the image of we are going to talk later in this video. This is the saffron in stain. It is the fourth solution or fourth reagent we can in the gram staining procedure. It's quite reddish or orange in color, reddish orange in real life, and yeah, it is been used as a fourth reagent. And after using acetone and using tap water, you have to go with saffron in solution and placed on the smear. Now you have to keep for 30 seconds, then erase the solution via use of tap water near sink. So this all are the stains, four stains, and this is the image of smear microscopy. We are going to talk about it. So this is the rack, this is the stain, Lugol's, and this is the crystal violet stain. Yeah. Now moving forward to and continuing the procedure way of this gram staining. So please watch till the end. You will find the smear microscopy explanation also.
The four solution or four agents. So this is the slide. This is a very little, okay, but we will magnify it and produce here for you to for ease to see it. Okay, so here, for example, it is a smear. Okay, at the one end of the slide. So at the one end of the slide you see the smear. So first first procedure, what you will perform, you will put this slide onto the rack. That's the first point. On rack, okay, on rack which are parallel, not, and you will put the slide, okay, with smear at one end. Okay, second point. Now what you will do, you will put this violent stain, crystal violet stain, on slide and cover all the smears. So covering the whole the smear with the crystal violet stain. Okay, completely. Okay, completely for 5 minutes. So after adding the violet stain, crystal violet stain for the whole slide, and keep the slide on the rack for 5 minutes. You have to keep the slide for 5 minutes on the rack, okay? So don't touch it, just leave and relax, okay? For 5 minutes you have to keep it for 5 minutes, okay? Now after that, after 5 minutes, what you have to do, you have to take a forceps, okay? So after taking a forceps, you have to grasp one end of this slide. So the slide, you don't have to put horizontal, you don't have to put vertical, you have to put at an angle, so there will be slope of slide. For example, this is the forceps and this is the slide. So I am grasping this with one end of the slide, so it is making an angle, causing a sloping edge, okay? So for example, our smear is here or here, okay? So at one end of the slide there is smear, so don't grasp the forceps where the smear is present, okay? So you have to grasp the forceps, grasp the forceps of the you have to grasp the forceps, and you have to use the side where the smear is not present, so you will not damage the smear side, so you can visualize better, okay? So for example, here is smear, so I will use this side, so here will be the sloping edge and here is the smear. So what I will do, I will put iodine solution from here, so it will go rolling down, okay? So we will touch smear, so iodine violet crystal precipitate will occur, and yeah, that will work for us. And what will occur, iodine, because of this iodine violet crystal will be seen, precipitates will be there. And now after this sloping edge slide, this is sloping edge, you have put here crystal iodine solution. I am representing with the black color iodine solution, and here is the smear, okay? Now fourth step, after this, put this slide back on the rack. Now you have to add iodine solution, this iodine solution only, this only. Now put the iodine solution again, which is fresh, not damaged, or means not used one, okay? So you have to put a iodine solution which is fresh, and put this iodine solution again on this slide, which is placed horizontally on the rack. Now for 2 minutes you have to wait and relax. Wait for 2 minutes after adding the iodine solution on the slide, which is placed on the rack near the sink, okay? So after 2 minutes you have to add a decolorizer agent called acetone. Use acetone. For using acetone, we have to first of all grasp the slide with a forceps again, and we have to make an angle like earlier we did for the iodine. So again, grasp a slide with another end where the smear is not present. For example, you are using this end. Just imagine this slide. Now add acetone for 2 to 3 seconds. Please remember, it's very important to understand, not for minutes, not for more seconds, just for 2 to 3 seconds. So you have to add for acetone for 2 to 3 seconds, so it will decolorize the solution of iodine and precipitates are present in the slide, okay? So acetone will work as a decolorizer, okay? Just remember that for only 2 to 3 seconds you have to add acetone, and then you have to wash the acetone placed slide with tap water. So acetone for 2 to 3 seconds on sloping slide, okay? After that, use tap water to erase this acetone, okay? To erase this acetone. Now what you will do, after this tap water you will add counter strain saffron solution. So saffron solution you will be used, and put the slide on the rack horizontally on the rack for 30 seconds. Okay, so after this 30 seconds of saffron solution placement, you have to wash the saffron solution with tap water. Okay, okay, so running tap water you can use, but gently, gently, not too fast or not with too slow. You have to place the thumb. You can use also thumb. When you are using a thumb on the slide, just make sure that you don't touch the smear of the slide, and you can use like this. Okay, see, this is my smear, and this is the end where the smear is not there. So my thumb at the other end, so I am putting the solution on my thumb, not on the slide directly. Okay, sorry, tap water on the thumb, not solution. The tap water will be running from my thumb gently to the slide where the smear is there. Okay, so it will wash the saffron containing solution on the slide. So the saffron containing solution will be gone, and so we have done all the procedure containing the four agents we have placed on the slide, which is kept on rack. Now finally you have to now finally you have to go with blotting paper. So with gently pressing the slide with a blotting paper, white blotting paper, on which there is no dust or any other particles, you have to gently press the slide. Make sure that you don't disturb the smear. Okay, and gently press the slide by blotting paper. Blotting paper. Blotting. BLOTTING. Blotting paper. And the 10th final step, by using a flame, you have to dry the slide. First of all the agents you have done, all the ninth point you are gone far. Now you have to dry the slide. You have to dry the slide with the help of flame, but gently, for some minutes, 1 to 2 minutes or that. Then you have to dry the slide and then put an oil. Okay, then put an oil on the slide, because we have to visualize on the microscope. So we will say that smear microscopy, that is by with the help of oil immersion lens. Okay, so we will introduce one single drop of oil on slide. That point I think oil, I remember, forget the name of the oil, but we have to put one drop on oil on this slide. Now with the help of oil immersion lens, you can visualize the slide. Okay, now what you will find on the slide with the help of microscopy, okay, that is called smear microscopy. And let's move at that topic. I hope you understand is this whole procedure of gram staining. It is help to distinguish between gram positive and gram negative. And now we will move towards what will be the results of gram staining and what you will see in gram staining. So you can click a screenshot of this by pausing the video, or you can move forward if you remember that thing. It is a very short thing. I have not gone into a depth thing, but main thing is that we will use these four agents. You can remember this thing: violet stain, iodine solution, acetone saffron solution. This is decolorization, this is counter stain, and this is main stain. Okay, okay, now we will rub this. Okay, now what you will see in gram staining by smear microscopy. Okay, you have to ask the question, why what you will see, so basically you will see this. Okay, basically see I have four colors. I don't have purple and pink color. So what I will do, I will represent the pink color with red. This is red color. Suppose understand that red color is pink color. So this is pink, light pink. Okay, both side. This is light pink. Okay, this is the background. Now I am representing the purple color. The purple color with with the this blue color. Okay, I don't have the purple color just now, so I will represent via blue color. Here you will do clusters. There will be clusters. Okay, okay, there will be clusters, because we know that only bacteria is not possible, that you will see only bacteria on smear microscope. There will be also presence of pus cells. Okay, there will be pus cells. Okay, there will be also pus cells. So this is pus cells, this is clusters. And previous video, if you have seen that the clusters presence indicates it is staphylococcus. Staphylococcus. But by seeing clusters, you cannot say which species of staphylococcus it is present. Okay, so the clusters that you have seen, this is the pus cells, pus, pus cells. So this is smear microscopy done via oil immersion lens after gram staining. Okay, this is purple color. I will write also here. This purple color, colonies. Okay, this is pus cells, staphylococcus. Okay, here it can also be a mixed culture or mixed slide. Okay, there can also be presence of other species or other organism. It can be, you can also see sometimes. Can be chains. What you will see this. Okay, sometimes it is mixed or sometimes it is single. Okay, so this, if you see single handedly this chains or pairs, then you can say it's streptococci. Okay, it is also gram positive, it is also gram positive, gram positive, gram positive. It depends what smear you have produced. If there is only presence of staphylococcus, you will see only clusters, there will be no chains. But nowadays you can also see mixed, gram positive, both gram positive, but presence of other organism also. Now what you will see if it's negative, if the gram staining is negative. So I am representing the pink with red. Sometimes you see bacilli, bacilli. This bacilli, pink color bacilli, bacilli. It's a pink color. So what I have said earlier, on smear microscope, it's a gram negative. It's pink color bacilli. Gram positive and gram negative both contain bacilli. It does not mean that it don't have the bacilli in gram positive. It also have bacilli, but the color is pink here. The color is pink here. It's not purple color or slight purple color. We have not seen a purple color. It's a pink color. After gram staining on the smear microscopy with the help of oil immersion lens, we are getting a pink color. So it's a gram negative organism. So it's a gram negative organism with bacilli. I hope you understand this smear microscopy. It's very important. Okay. Just take a good look and take a picture if you want, take a screenshot. Okay, so we are rubbing this and moving forward.
So at last, after getting results from oil immersion lens via microscopy, we are seeing this either of these two, not both of this. So if you see pink color, if you see pink color, rods, rods or bacilli or cocci, if it's pink color and it's quite slight brighter than background pink color, and you visualize this pink color with bacilli cocci or any other rod, then it's a gram negative. But if it's on the right side, you can see that if it's in clusters or in chains or in pairs and the color is not pink but it's purple, then it's gram positive. So these are the images or results we get after doing oil immersion smear microscopy with the explanation I given earlier about whole procedure of gram staining. So these are the results we get by doing smear microscopy, and it's very important for us to do this, to take a part in it and to differentiate whether it's gram negative or gram positive. So gram staining is the initial method to detect which bacteria has affected the human body or which infection has led to the symptoms or signs of the patient. So that's all. This is the gram staining video. That's the whole video you need to see, and I have given almost all the information about gram gram staining, and the image is taken from this website Adam, and they have many more images. You can visit it if you want. I will give the link below in description. And if you need any kind of other videos related to gram stain, please comment down in the video, and hope you guys like it. Please like, subscribe and share the videos to your colleagues or to the ones who don't know how to perform gram stain. Okay, so hope I will see you all guys in the next video. Till now, like, subscribe and share this video, and hope I entertained you by this giving this knowledge, and hope so you would do good in your studies, and stay tuned. All the best.